and they can get fragmented, and thereby the dynamics at the neuronal interface get perturbed. Can we start?
Yeah.
Thank you so much. So the next part is I talked about till now electrophysiology until now. Now I'm just taking you to some basic enzymology once again. Lactate dehydrogenase. Lactate dehydrogenase and post-translational modifications, which Dr. Pradeep was very interested in yesterday. So now I would, and in this talk, after this talk, maybe around in the middle part of the talk, I mean, the later part of the talk, I would like to come to the general winding up, and the last part also I'll take some examples, and then I'll try to wind up I'll try to get a holistic picture of the overall Murburn proposals. So LDH or lactate dehydrogenase is a ubiquitous glycolytic enzyme, and it's a clinical marker for several pathologies. All right, and Warburg effect is something that is it's also seen in plants. It's not to do with lactate. Some other photosynthesis also. So basically, high lactate in presence of oxygen. The classical theory states that lactate is formed in the absence of oxygen. When you exert the glycolysis pathway, ends in pyruvate, and the pyruvate is supposed to get converted to lactate, get reduced basically to lactate, and that is occurring usually in the absence of O2. When in cancer cells or in some other cells, when you have lactate production in the presence of oxygen, it's called Warburg effect. So it's involved in that also. So relevance in lactic acidosis, then lactic acid, the production process, it's very interesting. I will not go into details of this aspect. This was a very controversial aspect. That is, lactic acid production is also associated with acidosis. So there's some groups say there's no acidosis, it's not associated with lactic acid, and things like that. So I have addressed this in the paper, but I don't think we'll have time to address it in the discussion. I'm sorry I missed your question. We'll talk about your question. Okay, sorry. Then Cori cycle. Sorry, the Cori cycle spelling is wrong. CORI. So Cori cycle, if you remember, they got a Nobel. That is the lactic acid being moved to liver, and it's cycling from back. The carbons are used in the liver like that. So that's the relevance of LDH. Now let me come to what LDH is. What does its function? What is its function? LDH is a tetramer. Generally, lactate dehydrogenase means it's an oxidoreductase, but per se it does not have a redox center, in the sense it uses NADH or NADH or NADPH. NADH, correct. Because I work in so many systems, they have some have NADH and NADH. So here is the basic cycle of lactate involvement. Okay, see, lactate dehydrogenase is a tetramer. Here, the blue is A monomer 4 times, and this orange, yeah, this is in heart. You have all the 4 monomers are made of the B type. So LDH1 is predominant in the heart, made of tetrameric B. Muscles have tetrameric A, LDH5. So that, don't get too worried about the nomenclature. So they are also called muscle and heart, LDH2. So that's what it is. So the difference, there are so many multimeric LDHs which have 1 of this and 3 of that. That's not our concern. Classical perceptions being that A and B isocyans work in opposite ways. A works in one way and B works in another. It's supposed to explain why heart has little lactate production. It can continuously pump without lactic acid production, and muscle has the other. So now here is the the terminology forward and forward has to do with the name lactate dehydrogenase. If it is dehydrogenase, it's forward, and if it is the other one, it is backward. So LDHA, which is found in muscles, that is called the backward reaction. The LDHB forward reaction. So here is the classical modality of understanding inhibitions. That is, the traditional substrates are either blocked by the active site getting blocked like this, or at the outside. Did you get the topological idea of it? Basically, there's a deep channel in lactate dehydrogenase monomer, and this can get blocked by something that can go in and block it at the bottom, or something that can block it on the surface. This is the general perception. And also there can be some allosteric binding, wherein the tetramer might have something binding here between the between the monomers, and thereby messing with the active site conformation, and then this inhibition can also occur. So this is the overall schema of interpretation of lactate dehydrogenase function in the purview of classical physiology. So the classical physiology also deems that the fully reversible, the reactions are fully reversible. If so, the question is, why do A and B work out in opposite direction? Yeah, it does both. They also think that the mechanism is fully reversible. The code reaction is LDH binds to the nicotinamide moiety, then lactate, it makes lactate nicotinamide, then pyruvate second step, then gets protonated, and this one. And when it works the other way around, the forward or anti-clockwise ways, it works this way. The sequential exactly is the same order, and the intermediates have been so-called studied well in characteristics. Now, the point is that theoretical free energy change from redox potentials was calculated to be minus 26 kilojoules per mole, and that is the LDH utilization reaction. And from the equilibrium constant, it is minus 66 kilojoules per mole. All right. So the reaction that utilizes the LDH is thermodynamically facile. But then there was significant disagreement in theory itself between the values of the free energy change, calculated in two different ways. One this way, one that way. So now critical analysis comes directly. My model comes here. I have proposed a completely different model. I said that it is not that way. It is in a the forward and reverse reaction occur in two different ways, and there are also other things going on. So I will come to this model later, and now I am coming to the critical aspect of it. The critical aspect of it is now hydroxy acids. Yeah.
What I'm saying is that the theoretical, whatever you got that energy values, are those reported in any article?
Yes, they are reported.
And then have they performed quantum chemical calculation for that?
No, this is not quantum chemical. Those two.
How they like it?
No. Here it is by basic delta G is equal to NF delta E. You start with the redox potentials of the these things are known, and then you can easily, this is the experimentally determined equilibrium constant. From that you can determine. This is the from the electrochemistry values, redox potentials you can calculate. These two things are, there are three ways of calculating the one is delta H and T delta S, one that one, and the third one is this. What I do with my research collaborator is we start from the empirical determination of delta G of formation. It's a very simple method. I will come to that. All right, I'll show you that. One second. The ab initio calculations. No, we are not going there. That's too sophisticated. Okay, let's deal with this. So that's a good question, though. So here is the general equation of a hydroxy acid. Like these are the reduced hydroxy acids. Very important aspect in all biochemistry. One carbon, two carbon, three carbon, four carbon, 5 carbon, and 6 carbon. They form very important metabolic junctions in routine biochemistry. They are, say, glycolate, lactate, malate, hydroxyglutarate, isocitrate. So they are, and then the oxidized form is glycolate, pyruvate, oxaloacetate, ketoglutarate, and oxalosuccinate. So this is the reduced form. This is the keto acid form, hydroxy acid form, keto acid form. Now, if you calculate the free energy change, the reaction has very, very, very high free energy change value. Now, how can this be freely reversible? It cannot be. That's my argument. You cannot have such a reaction freely reversible. There is this this value becomes like this because of the involvement of proton. Proton in this contributes around 450 kilojoules per mole component. So this aspect of the proton contributing significantly in the overall thermodynamics was highly overlooked, which is why we had Peter Mitchell proposing the proton pump and all that. They had not really calculated that proton will contribute that much. Because when this gets into the equation, the OH bond formation comes in, and then that offsets lot many other things. So if you don't, then that is the reason that you had these kind of values. All right. So we will not get there. So fundamentally, the calculation I did the calculation for all these and showed that the delta G, it's not freely reversible, that the equilibrium constant is highly displaced. The classical purview of metabolism deems that most of the enzymatic reactions are around equilibrium and very small values here. So they say the assumption is that they are fully and freely reversible. But then, if that is the case, how would you explain how cells get powered? There has to be some source of energy input. If everything is around that, it doesn't. So there is where the context of Murburn comes. All right. Now, the second aspect wherein I criticize the classical model is, you look at the active structure. This is something that you structural biologists will love. There is practically zilch difference in the active site structure of human A and B active sites. This is the overall protein here, and this is the look at the deep where it binds. Hardly any difference between A and B. There is one isoleucine or something small difference here and there. One amino acid somewhere different, but that is not little topography change in amino acid residues. Binding based logic, there is hardly any difference. So Murburn. So the point that I missed here, I'm sorry, here is LDH5, which is found in muscle, is 4 times that found in liver. All right, the same isomer, the same tetramer, same isomer found in both muscles and liver. Then why does the lactic acid that is produced in the muscle get transported to liver? That's the question. I'm sorry, I missed the most fundamental question. It cannot explain physiology because both liver and muscle have the same isomer. Then how does the classical purview, which says that, you know, the B metabolizes lactate and A metabolizes, and then both are freely reversible? It doesn't make sense. All right. So that is the primary reason that we should fundamentally question, because if that is the case, liver also has the same isomer. Then how can we explain? So here is where Murburn model comes. Murburn model, I'll come to this later. It says that Murburn model says that the actual reaction between this is pyruvate and sorry, this is pyruvate making lactate and NAD plus, and this reaction that uses the NADH, because the proton comes this side, the 452 kilojoules, they offsets, and this you have highly favorable. So that means the pyruvate to lactate conversion is highly favorable in the cells, in the muscles. That can be catalyzed by the enzyme, but the reverse reaction cannot be catalyzed in physiological conditions
by the same enzyme. In the liver, what happens is, I told you in the yesterday's talk what was happening in liver. Cytochrome P450s. What do cytochrome P450s and the flavoenzymes entail? Reactive oxygen species. So you involve an oxygen into this reaction. You involve some other molecule also, and then redox reaction among them. That offsets the energetic cost. That is still highly favored. That this is the thermodynamic treatment. Here I will show you what the overview is. Here the modified backward, that is when pyruvate gets converted to lactate, the reaction, pyruvate goes into the active site, pyruvate binds to the positively charged area, and then NADH comes, and then this reaction occurs. And in the forward reaction, what happens is, once again, superoxide gets stabilized because of the lysine and arginine here. NAD plus binds in another orientation here, and lactate is bound at an alternate site. Did you understand the difference between the two models? The forward reaction, that is conversion of lactate to pyruvate, which occurs in the liver, requires oxygen-based mechanism. It is using the reactive oxygen species because that is thermodynamically feasible, that is allowed by the structure, and that explains the physiological premise. The current premise that that all physiological reactions, all enzymatic reactions are freely reversible. No, thermodynamics does govern kinetics. If at a most people think that, oh, delta G might be positive, but kinetically highly viable. Yeah, hoga. You have to move the product. You have to somehow enable that conditions to be met. So otherwise, the equilibrium constant is not favorable. So you can still have if you provide enough activation energy, but that's different. Now the point is here. You see, this is the model, and this is the structural conformation we talk. We get lactate binding here. It's not a very great in the sense lactate being a small 3-carbon molecule. It's not something that structural biologists will greatly appreciate. But the point is that the nicotinamide binding is fully supported. So you see why it goes to liver is justified by this simple explanation. And this also agrees with the structural aspect that there is no real demarcation in the active site. Now, how I'm going to justify that? The Murburn model is like this. Pyruvate comes and binds, and then there is a slight charge here, positive, it enables this binding. Then what happens is you have nicotinamide getting converted to the nicotinamide gets oxidized, and then you have the lactate pyruvate getting reduced, and then the lactate gets released. The classical model, if you see, it had what binding first? It had the NAD plus binding first. If NAD plus binds first, the lactate cannot enter the cell. So that doesn't make sense. So that's what I will justify the structural data. And then the forward cycle is, it involves ROS stabilization and then attack on the lactate by a stochastic mechanism. That's what is shown. Then this could also happen parallelly, that NADH and singlet oxygen could also get involved, and you have peroxide formation and all that here. So all these reactions are stochastically feasible, and that's why I have written that. So both forward and reverse reactions are altered from the classical pathway. Stochastic bimolecular interaction draws dynamics with positively charged residues is what I'm vouching for. So we have docked with both LDHA and LDHB, and we get similar results. So the NADH supposed to be bound to LDHB. You see correct orientation. You have the nicotinamide moiety going in and the adenosine moiety coming out. Whereas if it bound the same, the nicotinamide moiety, the NAD plus suppose substrate bound to NAD LDHB, you see it comes, the nicotinamide comes out rather than that one. So that's because the positively charged here will repel the positively nicotinamide. So this is one support. The same thing you see in LDHA also. So that's what is shown here. Basically, if you expect the classical theory to be operative, we would not get such good binding based data. So, yeah, my student. Yes. Flexible. Here, in the autodock, the protein is a rigid molecule. We can rotate some sp3 bonds of the ligand, but the protein is more or less rigid.
No.
Yeah, you can also do a little bit. That is for sure. That is there.
1 minute. 1 minute. 1 minute. You are only showing LDHB. LDH exists in two isoforms. One is LDHA4 and also LDHB4. LDHA4 actually is the one which will be converting pyruvate to lactic acid. Well, there are several other problems. I am not getting into it. But LDHB is supposed to actually convert lactate to pyruvate, right? So
Yeah.
this is supposed to be the heart muscle enzyme, and that is supposed to be actually skeletal muscle enzyme. Both are actually errors created by actually the 1922 Nobel Prize, but subsequently Meyerhoff and others have changed. Warburg did not change, is another matter. So, what is the structure of LDHA tetramer? How is the binding of this NAD and NADH to those things? Did you examine that?
Yeah, that's what is shown here, Professor Varlakonda. That's what is shown here. Both A and B are bound, and that's what is shown here. So the nicotinamide.
LDHA
basic-
structure, does it differ from LDHB structure or not?
No, there is no. Practically, there is only one small amino acid difference. I have said that there is no difference practically. So basically, but then in the active site there is no difference, but in the periphery there is difference. All right. Now I'm coming to that. Just like now diversity of LDH modulators. If you see, LDH is supposedly modified by one second. You see the case of Murburn concept. When you have diverse substrates, diverse modulators, there is where Murburn comes in, particularly in redox reaction. You understand? If you have such systems, you can very clearly understand that Murburn concept is involved because non-specific electron giving and taking is occurring. That's the case where Murburn gets active. That's the case where Murburn concept needs to be applied. You cannot have these kind of diverse moieties getting activation, inhibition of the same molecule. So now here you LDHA and LDHB, you can see the different molecules bind at different sites, and therefore you can explain some of the activations and inhibitions thereof. The topology is different, not the active site. Did you get what I'm saying? The overall picture. I do agree that the amino acids can swing, change, and thereby the induced effect can occur. I've just considered the native, steady, isolated X-ray crystal structure. That's all I'm considering. Assuming that that is the general structure, doesn't change much. This can be explained at least for the NADP, NAD plus, and NADH structures, and it explains beautifully. And my theory fits with the physiological function also. So here I have explained the isomers, and here is something our Professor Varlakonda will just jump into, I'm sure. Here is the normal versus cancer cells. All right, the metabolism. Here I'm not going to speak much. Just one thing. Here is the stoichiometry of acid and the conversion in the normal and the cancer cells. This is the normal cell. This is a cancer cell. What really happens is here there is also mitochondrial conversion. Now, when lactate get converted in mitochondria, it gets once again Murburn, right? Mitochondria is a seat of Murburn, because Murburn, lots of ROS in mitochondria also. So there also this lactate can get converted. So here the the rate at which cancer cells metabolize is very high. And they also produce lactate highly in presence of oxygen. Now, how is that happening? That there is a high amount of cytoplasmic Murburn also. So here, that's what I've shown here. The pyruvate and acetate formation in this is high, and lot of transport here. So this is the reason why you have higher amount of growth and metabolism in cancer cells. So what I propose is, can we approach cancer in a very subtly different manner, in the sense that if we understand the Murburn process properly, can we burn out the cancer cells more effectively? If you give molecules that can speed up this process. That's my suggested approach. I don't know, it's just a theoretical idea I had. So this is the difference, this is it. So that's it.
Generating more amount of hardware.
Correct. See, now I want to.
That also will take place.
No, that's what I'm saying. Now, very good question. See, now, if you came in my first talk, you would have seen I talked about azide increasing some catalysis, right? And then I showed in several cases, it depends upon the immediate ambience, all that. What I'm saying is that my student isolated from phenyl hydrazine something that could enhance the same reaction 25 times. So my concern is, my idea is this much, simple idea, that cancer cells have cytoplasmic Murburn high. So if you could see, I showed in my earlier talks, when the derivatized with in the cytoplasm, when the reaction is in the first phase, the different molecules, all that, there is definitely distinct effect. So if you can isolate some molecules or species of drugs which are needed at very small amounts, they can perhaps be used to attack the cytoplasmic Murburn more effectively. That's all I'm saying. There is a probability. There's a chance. What I'm saying is, Murburn occurs in a systematic manner everywhere. But these guys rely more on that in a soluble phase also. So rather than trying to minimize ROS and all that outlook for this, rather than that, you just burn it up. Give molecules that can specifically increase the metabolism that way. That metabolism, that reaction setup. It's just an idea. It's just an idea. And it can work based on my thinking of it. So the next aspect is modification of proteins, metaboloproteomics. The proteomics, see, here is the bigger picture. Murburn in cellular milieu, oxygen, triplet oxygen. In presence of heme, it can go singlet oxygen with sufficient energy, whether it is light, or it can, while going through this process also, it can produce singlet oxygen. Both singlet and triplet can go to superoxide. Superoxide can receive electrons and protons and go to hydrogen peroxide. And then hydrogen peroxide can, once gets an electron, can break down into hydroxide ion and hydroxyl radical. Then hydroxide ion gains an electron. Sorry, this is bad. Hydroxide ion, this is wrong. I should not put an E here. Sorry, this gets a proton and it becomes water. Sorry, that's wrong. So we know that there are other reactions involving all this dismutase reaction, catalase reactions, Haber-Weiss reaction involving and Penton reaction. So these are the normal, well-known reactions. Now you will ask, why Murburn concept? What is this? You're just recycling what is already known. Yes, all this together, you will call, suppose this is an amino acid residue like tyrosine, it can undergo reaction with so many of these ROS agents, that is singlet oxygen, hydroxyl radical, then superoxide, giving different kinds of products. So now in a milieu which has any single ROS, anything can change their, it's not easy to say I have superoxide, so only superoxide. When superoxide and heme is present, you will easily get peroxide, hydroxyl radical, and singular toxin also. All those are very easily, in equilibrium. So, in the Murburn purview, cellular medium must process diverse DRS that are produced. DRS exists in intricate interconverting equilibriums depending critically on pH. It is inevitable that diverse protein residues react and get modified according to this. Now, currently, yes, there is non-enzymatic PTMs known, but they are not seen in the context of generally favorable. So, here is something that if you are working on biological aspects, you might want to really understand and know, that is, in presence of a murzyme which can generate a ROS or modulate any ROS in presence of ions and metabolites, can auto model derivatize and also allo derivatize. It can post-translational modification it can do on itself and also on others. There is no necessity for kinases to do phosphorylations. There is no necessity for glycases to do glycosylations. This is the logic of that. Very simple. You see, this is how a phosphate group can be changed. Phosphate attacks, makes hydroxide. Then you get serine phosphorylated.
So another thing is like what you are saying, that there is no need of phosphorylation or glycosylation. So are you
saying that? You got me wrong. I'm saying there is no need for that enzyme. Yes, correct. What could happen is a random stochastic chemistry can also give that. That's what. Here is the ion. Here is the metabolite. For example, glycosyl residue. Any ion or any metabolite can get derivatized onto the protein. So it will affect your proteomics and metabolic studies. Did you understand what I just said? So this is the logic of Murburn catalyzed post-translational modifications. So how do you differentiate Murburn post-translational modifications with classical enzyme post-translational modification? So when you have a large one like ubiquitation and lipidation, generally enzyme based. So this is also enzyme based, but Murburn processes. Cofactor insertion, proenzyme cleavage, activation, then the classical like phosphorylation, acylation, all that. Here, sometimes you might have small crevices and transmembrane loci, allosteric sites inside, all that, random sites also. This is what you can say that it is by Murburn. So you can also look at the mechanism and you can say that. So the larger picture is that, there is utilization, basic simple utilization, that is superoxide, it can get, there is generation, oxygen to ROS generation. Then these are the different modulations. So heme, iron sulfur center, arginine, lysine, plavin, these are the typical amino acid residues and cofactors that are involved. These are the dynamics of the species that are there. In the Murzone, Murzone is around the enzyme, where this can happen. And then you can have different ions or different metabolites that can do carbonylation, nitrosylation, alkylation, acylation, glycation, phosphorylation, oxidation, halogenation. So for sure, I wouldn't imagine there's any halogenation enzyme in our system other than murzyme. So phosphorylation, practically many kinases are phosphorylated. But then, do you know which post-translational modification is the highest in our cells? Phosphorylation. And not all of them are to be attributed to the kinases. For example, in the last talk, you saw which one? NKA, the transmembrane region, then you get phosphorylation. How can we explain it? Kinases cannot go into that region. So that can be explained only by this. So highest phosphorylation is perhaps because of Murzyme, not because of plastic. So paradigm shift, I wanted to discuss one more aspect here. So that's why I'm going quickly. The paradigm shift here is even non-redox cofactor containing proteins can be murzymes. I'm not dealt with complex Y, it's a murzyme. And so here, what was the murzyme? Lactate dehydrogenase, right? It doesn't have a this thing, but if you study the active site, lots of lysine and arginine in the positively charged site. And now, you structural biologists, one thing that I would request you to study, if possible, is now if there is ROS, evolution will also enable you to optimize the ROS traffic. So the structural this thing, amino acid location on the topography, people are always obsessed with the active site. The ROS dynamics around the protein will require the effective, traffic of ROS around the protein. So positive, negative charges relay like that. So the ROS can stochastically move in a particular direction. That can also happen. So then reactions are all not always at delta G, close to zero, reversible. Higher yield reactions are all rooted via Murburn. Reinterpreting post-translational modifications via Murburn draws routing along protein contours. That's what I just talked about. So now I am trying to give you a large picture. Only one talk remains. So before that talk commences, that will also have one application of the bacterial flagellar motor. Before that, what is the most salient feature of life? Actually, I should have had this as an interactive one. It would have, a living being has the ability to preserve itself by commissioning non-spontaneous actions, right? That's what you would. Then what is the most essential requisite for the above? A simple powering modality interconnected with coherence, homeostasis, and response principles. You need a powering to and connecting of the different aspects, coherence and homeostasis principles, response principles. Does the cell theory or central dogma provide a tangible means for achieving the above non-intelligently and at short time scales? Did you understand the question? The question is, can the cell control its powering or the homeostasis or coherence aspects and sensing and responding without invoking, say, hormones or nuclear signals and all that quickly, immediately, at very, very short time scales, say in millisecond, microsecond, that kind of time scales, without the involvement of genetic control. There is hardly any principle like that. You know what is central dogma, right? What is central? Central dogma is genes give RNA, DNA, RNA, proteins, and then all these interactions are highly controlled on a topological basis. Yes, active site basis. And so the reactions resulting thereof, the proteins also interact with other molecules and macromolecules around them, high specificity and selectivity. So you have in cells reactions occurring only that are wanted, that are determined, that are destined to occur. Now, what about the stochastic events that can happen? You are using oxygen, crazy reactions can occur. Now, what I'm saying is cells have evolved to accept and adopt and use natural principles of chemistry and physics to aid their own survival, and it need not occur with only topological identification. I had said delta G of formation, the thermodynamic pull concept, control, reaction, and then partitioning effects. All these effects were not really considered as something that were controlling the outcomes. Now, with the new, what is the etiology expansion of hormone concept stems from mured closed burning. Burning is a redox process. The concept is a forthrightly derived idea or set of ideas starting from known facts. Why did I call it Murburn concept? Mured burning. It's just it's a concept. Oxygen is involved or may not be involved in anaerobic systems. It's just involved in some diffusible reactive species, radicals. So that's why I called it Murburn concept. How is Murburn concept relevant to life? Murburn concept conceives a cell as a simple chemical engine, performing useful work via the principle of effective charge separation brought about by the obligatory involvement of diffusively reactive species. Why is Murburn concept a fundamental principle of life? I'll come to this in the last one in more detail. Provides a non-intelligent stochastic framework of interactions between diverse molecules and ions that constitute the cell. In doing so, it enables a simple explanation for powering, homeostasis, coherence, signal transductions, electromechanical activities, etc. Till now, we have shown powering, homeostasis, coherence, signal transduction, all I've shown. Next one is electromechanical. So, what is the classical difference between classical and what is the salient difference? Classical perception primarily deemed diffusively reactive species as toxic waste products, whereas Murburn school deems DRS as quintessential elixir of life. This is very important. Does Murburn concept oppose or negate classical perceptions? Murburn concept is a larger set of ideas, including several aspects of the classical views, incorporating later known facts and improving upon earlier prevailing stigmatic and aesthetic perceptions. It is not opposing all the classical known facts. Enzymology, it doesn't say that classical enzymes bind at the active site with topology. No, I never questioned that. So, in quantitative perspectives, what is the impact of Murburn concept? Classical perceptions had purely deterministic schemes to account for the metabolic or physiological outcomes. In purely deterministic, in the sense, anybody can know that any molecule is supposed to be diffusing or moving around with under stochastic principles only. Brownian motion, diffusion, everything is occurring as a matter of stochastic phenomena only. But ultimately, something at the end of it, something happens. That process that happens is always deterministic. It is of a given finite stoichiometry, A plus B is equal to 2C or something like that. So now here is something there where you don't really have a stoichiometry, you don't have a finite route of reaction. You can have multiple routes coming and converging at the same time. So it accommodates stochastic events, thereby explaining non-integral stoichiometry in cellular process, contributors and routine function. So next part is I can take questions now here in this talk. I just did a survey so that the next part, when I finish the workshop, I want to ask you, did you get something, and what part was convincing, what was not so convincing, what was interesting, all that by the next time. So I given you a bird's eye view of what things are basically. Yeah. So, yeah. Yes, Anvesha. Yes, Anvesha. Please come here. Come here, and let people see. Let people come here. No, because this mic is here. Otherwise, I'll have to repeat what you say for the audience.
It's just listed out. I don't know.
Okay. Yeah.
So, do you know what percentage of oxygen that we inhale is actually used up by our body? Is it 100%?
Well, see, that's a good question. See, in most of these Murburn processes, these are equilibrium processes. Now, I showed you so many reactions. The reactions will also give oxygen. Sometimes, peroxide is formed from oxygen, then peroxide can go back to oxygen, or it can go to water. It depends. So, if you ask me, we know when we breathe in and the air we breathe out, much of it is mostly same. Only thing that increases is perhaps a little bit of carbon dioxide. So you mean to say the oxygen that is taken into the bloodstream, still there is in the arterial blood there is a high partial pressure of oxygen. Even in the venous blood there is some oxygen. I don't know the exact quantification. I should not beat around the bush too much. I should say I really don't know what. I don't know. Can that question be answered? Because that question the cell will always have some oxygen inside, and it's a dynamic process. I don't know how percentage of oxygen. I'm just trying to see if that question can be answered. what percentage of oxygen that we take in is used? I don't know.
I just did a search on
Yeah, what does it say? Yeah, what does it say?
this is from sharecare.com and I don't know, it says inhaled air is about 20%,
Yes,
tends to be the exhaled air is about 15% of
that's what I said. Mostly it is that. No,
this was about 5. I don't know.
yeah. I don't think that was a question, right? The one that is taken. Yeah.
Well, can
Anything
I
else?
can I enter?
No, sir. Yes, please. You're on.
Yeah, entire your last lecture. Before this thing, you have given me given a table of different metabolites which are impacting actually LDH function in last but third or fourth paper. List of before you started your Murburn system, you have given a table. I believe citric acid and other things are also there. Now, I am not talking about mutations of the protein. I am talking about the impact of other metabolites. I think before this thing, you have actually given the table where you have given actually different metabolites which are impacting actually LDH function. Can you go back?
These are the metabolites that are these are the molecules that are impacting LDH function.
No, can you go back? Can you go back to the table? Can you go?
This is not
No, beyond that. Still, you have a big table there.
no, this is the LDH talk beginning.
No, I think we are missing the table. You have given the table where actually citric acid and other metabolites also you have shown. Do they have impact on structure of LDH is another question which I wanted to ask. The second thing is the lactate dehydrogenase or lactate production in cells is biphasic. This was even in Meyerhoff first theory also. He has stated that are there actually citric acid and other molecule in this? You have. Okay. Can you expand it? Can you expand it? I am not able to see it actually. Can you enlarge the screen?
Yeah, okay. It will take me some time.
Yeah. Number one, with regard to your last lecture, lactate production inside the cells is biphasic. Number one is downstream of dihydroxyacetone phosphate, where lactate is also produced from different alcohol and also glycerol system, pyruvate and lactate are produced. That is where actually iron as well as chloride, and then this ROS also comes into the picture where the shift will be coming from anaerobic system to aerobic system. Under the aerobic system, lactic acid production is downstream of citrate, ATP citrate lyase, breaks actually citrate to carbon dioxide and then pyruvate. This pyruvate is the target of LDHA. And this pyruvate actually produces lactic acid. The moment lactic acid is produced by LDHA, it exits the cell, enters into the microenvironment, creates a lactic acidosis, where actually other molecules, you are not interested in that particular thing, but other molecules like the transcription factors which will be actually activating metastatic cells or proliferative cells will be activated in the microenvironment. So LDH lactate production is biphasic. One is downstream of dihydroxyacetone. The other one is lactate is a paracrine fuel which is circulated in the blood, enters into the cells. Metformin, the anti-diabetic drug, inhibits both entry and also exit of lactic acid. Entry is done through actually basicin type of molecule. exiting is done by ambient molecule which are tagged to what is called monocarboxylate transporters. This particular statement is important in analyzing this thing. Glycolytic lactic acid theory is a wrong assumption. Lactic acid is a paracrine fluid. Lactic acid is produced intracellularly also, but not from the pyruvate. It is from the dihydroxyacetone phosphate. If you want, I will show you one slide that I can show you that right on my table is there. If you want, show me, I will show you that particular slide.
So what Lakonda is saying is that there are two ways that lactic acid can be produced. The classical perception that lactic acid can be produced only by the pyruvate, reduction method. That's not the correct one. There is also another method wherein the lactic acid can be produced, and it's not wrong, not right to interpret the data based on only this kind of perspective. That's what Professor Lakonda says.
Even in the conventional models also, citrate synthesized, so-called TCA cycle, is exported outside. Citrate is lysed by ATP citrate layer. There acetyl-CoA is produced, and also malic acid is produced. Malic enzyme will break down actually malic acid to pyruvate, and pyruvate is reduced by LDHB to lactic acid. That is exported outside.
See, Professor Lakonda, I wanted to address the students now. LDH A and B. Now, did you understand one thing that I wanted to bring, one fundamental aspect I wanted to bring to your attention? That is, when you try to, what is the fundamental definition of an enzyme? An enzyme enables the achievement of equilibrium. It cannot do a magic, right? So LDHA can move this way, LDHB moves that way. No. The concentration terms, when the equilibrium constant is very close, when the concentration of terms are agreeable and the binding affinity of the enzyme is good, maybe there can be some isozymes which can have that distinguishing ability, but not this. Here, very clearly, the free energy change is too large, equilibrium constant is too large, the reaction kinetics will not permit, which is why lactate has to be moved to mitochondria or to liver. Did you get that message very clear? Yeah. So thank you. Yeah, we are moving on to the next topic because we are supposed to start at 12:30. Now it's already next one.